Good day, this is Professor Will, CBMG PhD, case Saturday, August 24, 2019. It is 2:49 and 1 second Eastern Standard Time, and this is a continuation of my pathophysiology research, so let's hop to it here. Damage. As a target for damage, DNA possesses a multitude of sites which differ in their receptiveness to modification. On a stereochemical level, nucleotides in the major groove are more receptive to modification than those in the minor groove. The terminal of DNA chains expose reactive groups, and some atoms of purine or pyrimidine are more susceptible than others. As a consequence, the structure of DNA represents a heterogeneous target in which certain nucleotide sequences also contribute to the susceptibility of DNA to genotoxic agents. The ability of cells to survive hostile environments is due in part to surveillance systems which recognize damaged sites in DNA and are capable of either reversing the damage or of removing damaged bases or nucleotides, generating sites which lead to a cascade of events restoring DNA to its original structural and biological integrity. Both endogenous and exogenous environmental agents can damage DNA. Many repair systems are regulated by the stressful effects of such damage, affecting the levels of responsible enzymes or by modifying their specificity. Repair enzymes appear to be the most highly conserved proteins, showing their important role throughout evolution. The enzyme systems can directly reverse the damage to form the normal purine and pyrimidine bases, or the modified bases can be removed together with surrounding bases through a succession of events involving nucleases, DNA polymerizing enzymes, polynucleotide ligases, which assist in restoring the biological and genetic integrity to DNA. Endogenous damage. Even at physiological pH values and temperature in the absence of extraneous events, the primary structure of DNA undergoes alteration. Many specific reactions directly influence the informational content as well as the integrity of DNA. Although the rate constants for many reactions are inherently low, it is because of the enormous size of DNA and its persistence in cellular life cycles that the accumulation of these changes can have significant long-term effects. Deamination, the hydrolytic conversion of adenine to hypoxanthine, guanine to xanthine, and cytosine to uracil-containing nucleotides is of sufficient magnitude to affect the informational content of DNA. Depurination, the glycosylic bonds linking guanine and nucleotides are especially sensitive to hydrolysis, more than the adenine and pyrimidine glycosylic links. The result is that apurinic or apyrimidemic sites are recognized by surveillance systems and as a consequence are repaired. Mismatched bases. During the course of DNA replication, there are non-complementary nucleotides which are incorrectly incorporated into DNA and manage to escape the editing functions of the DNA polymerases. The proper strand and the mismatched base are recognized and repaired. Metabolic damage. When thymine incorporation into DNA is limited either through restricted precursor UTP availability or through inhibition of the thymidate synthetase system, DUTP is utilized as a substitute for thymidine triphosphate. The presence of uracil is identified as a damaged site and acted on by repair processes. Oxygen damage. The production of oxygen superoxide or hydroxyl radicals as a metabolic consequence, as well as the oxidative reactions at inflammatory sites, cause sugar destruction, which eventually leads to strand breakage. Exogenous damage. The concept of DNA repair in biological systems arose from studies by photobiologists and radiobiologists, who studied the viability of mutagenicity in biological systems exposed to either ionizing or ultraviolet UV irradiation. Target theories derived from the random statistical nature of photon bombardment led to the identification of DNA as the primary target for the cytotoxicity and mutagenicity of ultraviolet light. In addition, most of the structural and regulatory genes controlling DNA repair in Escherichia coli were identified, facilitating the isolation and molecular characterization of the relevant enzymes and proteins. Ionizing radiation. The primary cellular effect of ionizing radiation is the radiolysis of water, which mainly generates hydroxyl radicals, HO. The hydroxyl radical is capable of abstracting proteins from the C4 position of the deoxyribose moiety of DNA, thereby labelizing the phosphodiester bonds and generating single and double-stranded breaks. The pyrimidine bases are also subject to HO addition reaction. Alkylation damage. This modification occurs on purine ring nitrogen's cytotoxic adducts at the O to the power of 6 position on the guanine end at the O4 positions of the pyrimidines mutagenic lesions and the oxygen residues of the phosphodiester bonds of the DNA backbone biologically silent. Alkylating agents are environmentally pervasive, arising indirectly from many foodstuffs and from automobile exhaust, in which internal combustion of atmospheric nitrogen results in the formation of nitrate and nitrites. Bulky adducts. Large bulky polycyclic aromatic hydrocarbon modification occurs primarily on the N2, N7, and C8 positions of guanine, invariably from the metabolic activation of these large hydrophobic uncharged macromolecules to their epoxide analog. Ultraviolet irradiation. Most of the UV photoproducts are chemically stable. Their recognition provided direct biochemical evidence for DNA repair. The major photoproducts are 5,6-cyclobutane dimers of neighboring pyrimidines intrastrand dimers, 6,4-pyrimidine-pyrimidone dimers, 6,4 adducts, and 5,6 water addition products of cytosine hydrate. And this is a glossary that concludes this topic. Endonuclease. Nuclease that hydrolyzes internal phosphodiester bonds. Excision. Removal of damaged nucleotides from incised nucleic acids. Exonuclease. Nuclease that hydrolyzes terminal phosphodiester bond. Glycosylase. Enzymes that hydrolyze N-glycosyl bonds linking purines and pyrimidines to carbohydrate components of nucleic acids. Incision. Endonucleic break and damaged nucleic acid. Ligation. Phosphodiester bond formation is the final stage in repair. Nuclease. Enzyme that hydrolyzes in the internucleotide phosphodiester bond and nucleic acid. Resynthesize. Polymerization of nucleotides into excised regions of damaged nucleic acid. Transcription. The synthesis of messenger and RNA from template DNA. Good day, this is Professor Will, CBMD PhD. Today, Saturday, August 24, 2019. It is 4:12. 0 seconds Eastern Standard Time. This concludes my reading on pathophysiology. Hope you enjoyed listening. Have a great day.